In this video, we will look at the interphase of cell cycle in detail. If you don't know what cell cycle is, I have given link of a video in the description below which is about cell cycle. So let's get started. Basically, interphase is the preparation phase of a cell for division. Now, in this phase, the cell completely prepares itself for division. It may be mitosis or meiosis. So, interphase is the period between end of one cell division and the start of the next division. In interphase, the nucleolus and the nuclear envelope are distinct, mean it's clearly visible under a microscope, and the chromosomes are in the form of thread-like chromatin network. Further, interphase is divided into subphases to understand better. Its subphases are G1 phase, S phase, and G2 phase. S phase means synthesis phase. We will look at each of these in detail. G1 phase. G stands for gap, so it means that it's a time duration in which some events occur. It's the longest subphase of interphase. It means that G1 phase is the longest phase from other two phases subphases of interphase, mean S phase and G2 phase. Cells grow in size and function normally, which means that in this phase the cells are metabolically active and prepare most of the ribosomes, RNA, and chemical changes occur, in which the cells grow in size, about double of its size. In this phase, the cell grow in size and high amount of proteins and enzyme synthesis occur and produce more organelles. In this phase, the cell prepares itself for DNA replication by creating ATP. The second phase is S phase. S means synthesis phase. In this phase, the DNA synthesis occur, which means that DNA replicate itself, but the number of chromosomes remains same. Suppose this is a DNA in the form of chromatid. Actually, in S phase, the DNA is not in this form. It's in thread-like chromatin, but actually, to understand, I show it in the form of chromatid. Suppose this is a one chromatid. It's replicate in synthesis phase and become double. This one and this one chromatid are called sister chromatid because these are the exact copy of each other, this one and this one. In this phase, the replication of centrioles also occur, and the cell continue its growth throughout the S phase, and protein and enzymes are also synthesized in this phase. The third one phase is G2 phase. G again stands for gap. G2 phase is the period between completion of DNA synthesis and the beginning of the active cell division. In this phase, the cell prepares protein required for the division, like spindle fibers. The next event occur in this phase is the movement of centrioles towards the opposite poles, which play an important role in division. Beside this, there is another phase which is called G0 phase. When the cell don't have to divide, it enters into a G0 phase. At first, it was thought that cells enter into a G0 phase because of environmental factors like lack of nutrients. But it have multiple reasons, because some of our body cells enter into a G0 phase permanently or semi-permanently without any environmental factor. Like the cells which enter permanently to the G0 phase without any environmental factors contain neurons, the brain cells, the central nervous system cells, and some of the cells of our body enter semi-permanently into a G0 phase, but they divide when they got signal, and these contain liver and kidney cells. But make sure that these cells which enter into a G0 phase are metabolically active. The cell in interphase look like this. Nucleolus and nuclear envelope are clearly visible under a microscope, and the chromosomes are in the thread-like chromatin network. The another most important function occurring in cell cycle is the checking of a cells if it have some errors, and these are carried out by a cell cycle checkpoints. Cell cycle checkpoints are control mechanism in the eukaryotic cell. Its function is to prevent the mutant cell, to prevent those cells which have some error. The major checkpoints are G1 checkpoint, G2, M checkpoint, spindle checkpoint. G1 checkpoint is also called restriction checkpoint. It is present at the end of the cell of G1 phase, and its function is to check the cells that are ready for DNA and centrosome duplication. G2 M checkpoint. It is also called DNA damage checkpoint. It is present at the end of the G2 phase, and its function is to check the DNA and prevent the mutant cells until the DNA is repaired. The last one is spindle checkpoint. It is present in metaphase of mitotic phase, and its function is to prevent the separation of chromosomes until each chromosome is attached properly to the spindle fibers. The interphase takes most of the time of the cell cycle. Thanks for watching this video. If you like the video, subscribe my channel and press the bell icon and stay updated.